I'll start it now.
All right. So this is the bread project with all of us. Yep. And the background of yeast, its domain is eukaryota. Its kingdom is fungi. Dry yeast species include Saccharomyces cerevisiae, and it's often used in making alcohol and baking, and grows on sugars like glucose, fructose, sucrose, and maltose. It can grow aerobically with oxygen and anaerobically without oxygen, as shown in the picture.
Yeast background continued. As facultative anaerobes, yeast can switch between aerobic and anaerobic respiration depending on the amounts of oxygen available. The two forms of anaerobic pathogens are fermentation, which undergoes glycolysis, and anaerobic respiration, which undergoes all three parts of cellular respiration but uses a different final receptor in the ETC in place of oxygen. Yeast in bread creates bubbles in the dough so that the bread comes out fluffy and airy after baking. The air pockets are created by the release of carbon dioxide, a byproduct of fermentation. In alcoholic beverages, the final product of fermentation is ethanol or alcohol, and brewers manipulate the amounts of sugar and oxygen to create the perfect environment for a beer or wine. Yeast alcoholic fermentation. In the absence of oxygen as an acceptor at the end of the ETC, yeast undergoes a process of alcoholic fermentation. Through glycolysis, glucose breaks down to form pyruvates, which are then reduced into acetaldehyde and ethanol. The pyruvates are unable to continue the process of aerobic cellular respiration since the NADH created in the glycolysis cannot drop off its electrons and become NAD plus again. So the NADH donates the electrons to the acetaldehyde to create ethanol and regenerate NAD plus. Acetaldehyde is created when the carboxyl group from a pyruvate is removed and releases carbon dioxide.
Gluten works with yeast during the fermentation process to trap bubbles of gas inside the bread for a chewy texture. Gluten development occurs when water is added to flour. Protease in wheat flour then cuts gluten into pieces that connect with each other to form chains, which give the bread dough the stretchy characteristic, otherwise known as a gluten network. And then this is our experiment. So our hypothesis was that the release of CO2 would change the volume of the bread. Less yeast would result in less or no rise in dough, and more yeast would result in more rise in dough. Our independent variable was the yeast, and the dependent variables were the volume.
Okay, so the variables that we controlled for were the arrest time of the dough, which was around 2 hours, the yeast amount, and the measurements, and also the volume. But we didn't control for the temperature or the touching of the bread, which could affect the yeast fermentation. So the baseline recipe was 3 1/4 cups of bread flour, 2 teaspoons instant yeast, 2 teaspoons coarse salt, and 1.5 cups of water. We let the dough rest for 2 hours, and our variable that we changed, Olivia had 0 teaspoons of yeast. Griffin had 1 teaspoon of yeast. Caroline had 3 teaspoons of yeast, and I had 2 teaspoons of yeast. And also 40 teaspoons of yeast. So for our data, we had Olivia had at 0 teaspoons, the initial volume changed by 0%. At 2 teaspoons, the initial volume changed from anywhere from 140% to 155%. For 3 teaspoons, the initial volume changed 181%. And for 4 teaspoons, the initial volume changed 432%. So this is our graph that shows how it increased based on what amount of yeast we put in. Olivia, I think it's your turn.
Oh, this is the image processes of the dough initially after it was just mixed. On the left side is the control, and the right side is 0 teaspoons, 3 teaspoons, and 4 teaspoons of yeast.
Is this slide 13?
I don't know, just go.
This is slide 15.
Oh, 15. So the image processes final dough after 2 hours is what it looked like. As you can see, the control and then the different variables, the 3 teaspoons and the 0 teaspoons.
This is the image processes of the bread after being baked. We don't have images for every part, but we have most of the pictures. For our qualitative,
I can do this one. So for our qualitative observations, oh wait, this one we all do together. My bad. You can go.
for the qualitative observations, for 0 teaspoons, the initial dough was very stringy and it was a little bit sticky. After resting, there was no bounce and it was still sticky. After being baked, it was hard. There was no rise and it looked kind of raw or uncooked with no like brown color on top. For the 2 teaspoon control, the initial dough was sticky, but also kind of stringy. The dough after being rest was bouncy, airy, and it looked like it doubled-ish. And after being baked, the bread was chewy, it was kind of salty, there were lots of air bubbles, and the top was brown.
For the 3 teaspoon experiment, the dough is very similar to the control. It was still sort of shaggy until it was worked together. And it was sticky in texture. After resting, it grew more laterally than vertically than the control recipe, but it was still overall pretty similar to the regular recipe. After baking, it was slightly tougher, and there was a more apparent yeast taste. And I think in the photos it was a bit darker in color, but that could have been due to the placement in the oven.
So for 4 teaspoons, the initial dough felt just like the control, very sticky, maybe a little bit drier than what the control was. The dough after rest grew a lot to almost fill the entire bowl, and bubbles were visible throughout the mixture. After baking, it had a pretty hard outer shell, but it was much more light and fluffy on the inside because of all the air bubbles.
Overall, the more yeast that was added to the recipe, the more volume the baked bread had. and as seen by the 0% volume increase when there is no yeast added, this is due to alcohol fermentation in the yeast cells. Yeast cells are fungi that can do anaerobic and aerobic respiration, as the cells can cycle NAD+ to NADH and vice versa. For the most part, alcohol fermentation, which is anaerobic respiration, is what causes the volume to increase, as the products of the reaction are carbon dioxide and ethanol, which get released in and captured by the dough and increases the dough's volume. Therefore, the more yeast, the more CO2 and ethanol released to increase the bread's volume. Our lovely sources here. And that's all.